Okay, thank you very much. So it will be a challenge to try to treat about biologic drugs, biosimilars, and PV concept in 30 minutes. But it is a Irene challenge, so I follow Irene. So we will try. So as an introduction, what is a biological drug? According to the FDA, biological products are diverse category of products produced through biotechnology in a living system such as a microorganism, plant cells, or animal cell. But according to the EMA, biological medicine is a medicine that contains one or more active substances made by or derived from a biological source. Currently, the biologicals represent 25% of the world drug market, and the therapeutic prospects are mainly oncology, inflammatory disease, HIV, infection, etc. Sorry, it's not extremely readable and legible, but you have two types of biologicals products. You have non-stemmed from recombinant DNA, like vaccines, hormonal products, blood-derived products, cellular therapy, and you have stemmed from recombinant DNA, like oligonucleotide, DNA vaccines, therapeutic protein with growth factor, hormones, cytokines, plasmatic factors, and you have also antibodies, gene therapies, and recombinant vaccines. What about the market? It's in a rapid and growing evolution because since they appeared, biological drugs represent more than 25% of the world drug business, and among the top 10 sold drugs, 7 are biological ones. And the large majority of the emergent drugs are now biological ones. So you have here resources from Lehman Brothers, showing the part of the biologicals, the growing part of the biologicals. And here you have the top 10 in 2017 of the most sold drugs. If we go more in depth, biologicals are complex and high molecular weight polypeptides. And I will show you the example of a monoclonal antibody and the aspirin. So you have here the insulin, the growth hormone, and the monoclonal antibody. So you see the different size, and among the biologicals, you have also a different size. So due to their heavy weights and peptidic structures, the biological products have specific structure space, whose primary, secondary, tertiary, and quaternary size and structure condition both efficacy and tolerance. So just a recall, but here you have the primary protein structures, which is the sequence of a chain of amino acid, the secondary protein structures with the local folding of the polypeptides chain in hélice or sheets, the tertiary protein structure with a three-dimensional folding pattern of a protein due to side chain interaction, and finally the quaternary protein structure with a protein consisting of more than one amino acid chain. So the tertiary and quaternary structures are provided by complex glycosylation that ensure both biochemical stabilization and modulation or effector function. It is an important process, glycosylation, to ensure and to guarantee the folding and the three-dimensional protein structure, to ensure stability, interaction with targeted receptor, and optimization of pharmacokinetics and dynamics parameter. And of course, it conditions the immunogenicity of a drug in the patient and the effectiveness or the inefficiency. I recall you that lack of efficacy is considered like a side effect. I give you here an example with the infliximab, which can be manufactured by the same process, but with 3 glycosylation types. You have here the 3 types of glycosylation profile, which condition, of course, the immunogenicity of infliximab. You can have fucosylation, high mannose, or galactosylation. So since the glycosylation adducts are relatively unstable and depend on the manufacturing process, of course, the main associated risk for the biological drugs is the potential for modification of the therapeutic or the tolerance profile due to physicochemical modification like aggregation, absorption on surfaces, hydrolysis, oxidation, etc. Due to their structures and their glycosylation profiles, biological compounds can differ between each other in a same batch. So why are there variabilities? I just summarize the manufacturing process. So you begin with the cloning into DNA vectors to transfer into a host cell with expression and cell development. Finally, you have the large-scale fermentation, downstreaming, and finally the formulation. But as biological medicine are made by living organisms which are naturally variables, the active substance in the final biological medicine can have an inherent degree of minor variability due to the glycosylation process. So, some word of regulation. Unlike chemical drugs, molecular identity cannot be established for any two biological drugs, even if they are synthesized by the same process. So the active substance in the final biological medicine can have an inherent degree of minor variability that we call microheterogeneity. This minor variability must fall within an acceptable range to ensure consistent safety and efficacy, and this is done by adjusting the manufacturing process to guarantee that the active substance fit into the desired specification range. And you have here what is acceptable of a biologics with 3 different batches, and you see that glycosylation can differ, but for many reasons it is considered to be acceptable. So according to ICH QE5, the demonstration of comparability does not necessarily mean that the quality attributes of the products are identical, but that they are highly similar and that existing knowledge is sufficiently predictive to ensure that any differences have no adverse impact upon safety or efficacy in the drug products. So the range of variability must be established for any biological products and for every step of the manufacturing process. Now the biosimilar concept. What is it? The EMA defines as biosimilar as the biological medicinal products that contains a version of the active substance of an already authorized original biological medicinal products, with similarity established in terms of quality characteristics, biological activity, safety, and efficacy based on a comprehensive comparability exercise. So a biosimilar doesn't need to be an exact, but a close enough copy of the original biological reference. And the concept is commonly summed up that it is similar but not the same. So I summarize the differences between a biosimilar and a generic. If we talk about the structure, chemically, for the biosimilar, it's not the same active compounds. For a generic, it is the same active compounds. The differences are accepted in a defined range for the biosimilar, but not accepted for the generic. In terms of development, for the biosimilar, you have non-clinical may not be required and clinical trial necessary, and it is the opposite for the generic. You don't need any clinical studies, but non-clinical study for PKPD, you need that for, to establish the bioequivalence. So development study results, it must be similar, but can differ in an acceptable range. Here it must be bioequivalent. The development is very long, even for a biosimilar, it's around 8 to 10 years. It's only 2 or 3 years for a generic. The development cost is quite elevated for a biosimilar, $100 to $200 million. But it's only $3 to $5 million for a generics. The discount you can expect is 15 to 30% for the first biosimilar, because usually more you have biosimilar, more the discounts will increase. But it can be very elevated for the generics with 50 to 90% discount. Interchangeability. For the FDA, it's possible to interchange, but I will come back on this concept with specific studies. And for the EMA, it is a national decision. So we are 27, nearly 26 now, and we can have a different policy. But interchangeability, it's since the approval for the generics. And finally, in terms of immunogenicity, biosimilars are usually immunogenic and non-immunogenic for the generic. Split this one, sorry. Okay, so in 2017, only 11 biosimilars were approved in the EU. So you have here the main products, but the marketing perspective shows that a lot of biosimilars are expected due to the patent cliff. And you have here in the triangle the best-selling drugs in the world. It is really a growing market. So in terms of structure for the biosimilar, it corresponds to biological reference with a different glycosylation profile due to different manufacturing process. But it could include minor differences in the stabilizer or the buffer compared to what is used in the reference product. So if you remind the first scheme, I show you, you have at the beginning the same amino acid sequence and maybe the same genetic sequence, but the DNA factor, you can have differences at the level of the DNA vector. You can have a different recombinant cells and expression system, a different fermentation process, a different downstreaming protocol, a different in process control, and maybe a different formulation. So, slight differences, of course acceptable within products variation, are expected during the manufacturing process for biological products, regardless of whether the product is a biosimilar or a reference product. And it is a limit of the concept of biosimilar. And a reference products and biosimilar batch-to-batch differences must be in the same range. So as mentioned by EMA, a natural variability is inherent to all biologics, and a strict control are always in place during the manufacturing to ensure that it does not affect the way that medicine works or its safety. So you have in mirror the development of a biosimilar and a reference product in terms of amount of data required. With a new biological development program, you need a lot of data in clinical phase, but finally few in the preclinical, and it's exactly the opposite if you go about biosimilar. So very briefly, the development and requirement of biosimilar. So first of all, in terms of analytical development, you have the easiest development process in terms of biosimilar because unlike with generics, the reference product is protected by manufacturing process patent. The second step concerning the non-clinical trials, it may not be required unless we can show clear differences in terms of process-related impurities and important differences in formulation. The third step concerning the PK/PD establishment, you must demonstrate PK/PD similarity and assess clinically meaningful differences. And it must support a demonstration biosimilarity with the assumption that similar exposure provides similar efficacy and safety. And the acceptance range is around from 80 to 125%. The fourth step concerning the clinical trial, it must design to investigate whether there are clinically meaningful differences. The goal is to support a demonstration of no clinically meaningful differences, and you have to consider the adequacy of population sample size and study duration to dissect the differences. And the fifth step is the risk management plan, which must cover the non-risk for the reference products and the theoretical ones of the biosimilar product. Just some word about a very interesting thing, which is a bio-better concept. Unlike biosimilar, the bio-better present a superiority in one or various aspects of their clinical profiles by structural changes or improved formulation. And it results in an improved safety and or efficacy and a decreased cost. So, according to the FDA, bio-betters are considered to have different active compounds, and it requires the same development biological reference, but the probability of regulatory approval is quite higher. And you have here example of bio-better development technique. For example, you can have pegylation, humanization with human glycylation patterns, or peptide addition enhancing half-life or the target function. Now, what are the main pharmacovigilance challenges in terms of biosimilar? So due to the intrinsic limitation of pre-marketing clinical trials and a low predictivity of animal study, due to the high immunogenicity potential and manufacturing variability over time in the post-authorization phase, the patent cliff and the increased biosimilar authorization and the moderate feedback, you see that you have many, many challenges. So due to you have challenges in terms of limitation of pre-marketing clinical trials and predictivity, because the animal study may not be required, you have necessity of relevant species, it's difficult sometimes to extrapolate, and finally, in pre-marketing clinical trials, you have an interesting limitation due to the product variability. If we consider the high immunogenicity potential, you have a multifactorial risk with patient factors, comorbidities, genetics, previous or concomitant drug exposure, etc., and product factors with the possibility of impurities, structural modification and protein aggregation, variability over time in the post-authorization phase of active substance. And so the biosimilar safety assessment must be considered on a drug-by-drug basis and based, of course, on traceability. 2 words about the interchangeability. According to the EMA, it refers to the possibility of exchanging one medicine for another medicine that is expected to have the same clinical effect. This could mean replacing a reference product with a biosimilar or vice versa, or replacing one's biosimilar with another. And we have here 2 clear different regulatory concepts. For the EMA, the decision of interchangeability is taken at national level and relies on national safety efficacy data and evaluation performed by the EMA scientific committees, mainly the PRAC, the Pharmacovigilance Risk Assessment Committee. But for the FDA, an interchangeable product is a biosimilar which has met additional regulatory standards relating to the interchangeability, including dedicated clinical switching studies. So, when you change, when you propose the patient to change, you have usually negative treatment expectation who are known to reduce a medicine effectiveness or increase side effects experienced by patient. So, as switching is generally carried out for cost-saving reasons, it could be responsible of safety assessment matter of issues, and it must really be taken into account. So it really must be considered for the biosimilar safety assessment. Remember that. Now, what are the advantages or the disadvantages of a biosimilar? So, of course, you have as an advantage is a lower drug cost, enhanced patient accessibility, around 54 billion estimated cost saving of biosimilar over 10 past year. But the disadvantage is that the interchangeability is difficult to reach. You have continuity of supplies, but product variability induced by manufacturing process is a constant risk. And of course, the last advantage is to provide extensive non-clinical and clinical data. So, as a conclusion, at the PV requirement, you need a strong monitoring due to biological drugs complexity. You need a new pharmacovigilance monitoring due drug-by-drug basis, complex dose effect, immunological adverse event requiring a rethink on the PV system. Because for the time being, we are using old concept to treat new drugs. And we are using the tool of the chemical drugs to assess the pharmacovigilance and the safety profile of the biologic drug. So we are here all together to rethink the system, because the pharmaceutical concept is exactly the opposite. It's absolutely not the same. And finally, the PV challenge to allowing to worldwide patients an access to safe, effective, and affordable, innovative therapeutic. So thank you very much for your attention.